Yes, yeah.
Well, good afternoon, everyone. Thank you all for being here. Thanks, especially to Jody for inviting me. The truth is that I would have loved to be there in Athens, but I couldn't manage very well my shared parenting, so I have to do the presentation online. Okay, so during the talk, I will mainly focus on our research on first-time mothers, but I will also dedicate a slide about our studies on fathers, but just to put into perspective the differences in magnitude between the two groups. Well, before proceeding, I think it's necessary to disclose that for simplicity in this presentation, I will use the term mother to refer to biological mothers that go through pregnancy, and the term fathers to refer to biological fathers whose partners go through pregnancy. Okay, so as you know, pregnancy is a period of extreme adaptations in the woman's physiology, and these adaptations not only affect the reproductive system, but also the cardiovascular system, the immune system, the musculoskeletal system, respiratory system, and so on and so forth. So in short, almost every organ of the mother's body adapts during this period to facilitate pregnancy, delivery, and postpartum. And recent neuroimaging studies in humans show that the structure and function of the human brain also changes during pregnancy. And these findings, together with the extensive literature in non-human animal models, positions pregnancy as a unique period of neuroplasticity in adult life. But what is neuroplasticity? So neuroplasticity is the intrinsic ability of the brain to adapt and respond in response to internal and external stimuli. And this is very important because that means that neuroplasticity can be driven not only by intrinsic biological factors, but also by external environmental demands. Neuroplasticity can involve changes at different scales, changes, for instance, in the proliferation and differentiation of different brain cells, not only neurons, but also glial cells. Also changes in the morphology and connectivity of these cells, as well as changes at the molecular level that affect the synaptic transmission between neurons. Well, one of the main mediators of neuroplasticity across life are hormones, especially sex-steroid hormones, such as estrogens and progestagens. For instance, during puberty and adolescence, the gradual increase in sex-steroid hormones coordinate a series of cellular adaptations, such as synaptic pruning and myelinization, that translate into macroscopic gray matter volume and decreases during the pubertal transition. But pregnancy is another period of surges in sex-steroid hormones that actually exceed any other human hormonal transition. And just to give you an example of the magnitude of these hormonal changes, during just the 9 months of pregnancy, the brain is exposed to an amount of estrogen equivalent to about 40 years of non-pregnancy. In fact, the similarities of pregnancy and adolescence at the hormonal, behavioral, and neural levels are such that scholars are starting to coin the term matrescence to refer to the process of becoming a mother. Still, while the brain changes in adolescence are subject to intense study, the neuroplasticity of pregnancy is still in its early years of research. So rodent studies, especially rat studies, have provided a great understanding of the link between hormones, brain, and behavior across pregnancy. In rats, for instance, late pregnancy hormones such as estradiol, prolactin, and oxytocin reach specific receptors in the brain to activate maternal behavior. More specifically, these hormones reach hypothalamic areas such as the medial preoptic area to do two main things. One is to inhibit the avoidance or aggression circuit, and also to activate the reward or motivational circuit. So once these two things has happened, that is, the first circuit is inhibited and the second one is activated, the animal can respond to the external stimuli of the pup, which at this point will be extremely rewarding for the mother, and shape that circuit and adapt it to its needs. So this circuit of maternal behavior can also be activated in virgin females and also in males, just by means of the continuous exposure with PAPS stimuli. The activation of this circuit involves a series of structural neuroplasticity changes that range from changes in protein expression and synaptic transmission to larger changes affecting the dendritic branching, dendritic spines, myelinization of axons, and also changes in neurogenesis and changes in the structure and number of glial cells. And for those who want to know more about these neuroplastic changes, I recommend you read the review written by the organizer of the symposium, Jody Pawlowski. Here you have the title in case you want to search and read carefully. Well, in humans, studying neuroplasticity is a little bit more complicated because we cannot analyze what happens at the cellular level, or at least it's not easy, not ethical either. So as a proxy, we use data from structural magnetic resonance imaging. So structural MRI not only provides a good resolution of the different brain tissues, we can see what happens at the gray matter, white matter, and CSF changes, but also allow us to parcellate the brain into different structures and see whether specific regions of the brain differ in different metrics. But what is really important, especially for this type of population we're studying here, which are mothers that go through pregnancy, is that MRI is considered a non-invasive technique. And that can be used under certain parameters, can be used while you're pregnant. Okay, so what do we know about the neural changes across pregnancy in human mothers? The question is, does pregnancy modify the brain? Is the brain of a nulliparous woman different to that of a mother? Is this brain here different to this one? So to answer this question, we performed a study that was published now 5 years ago. In this study, what we did was to scan 25 first-time mothers before and after their first pregnancy. Sorry, before their pregnancy, and then again at 2-3 months postpartum, and that was their first pregnancy. And we also scan control groups, a control group of nulliparous women at similar time intervals, as well as their respective couples that also serve as control. So we have also first-time fathers and non-father males. And I'm going to show you here the results. What we did was to compare the state of the brain here to that at this other time point. And we found that mothers display pronounced gray matter volume reductions from preconception to 2 months postpartum. And what is really interesting is that these reductions were not randomly located in the brain, but reductions were located in a region that clearly overlap with what we call the default mode network, here depicted in dark pink. And as you know, this default mode network is a large-scale functional network that is involved in social cognition, theory of mind, and also self-perception and mind wandering. Well, to find out whether the changes were limited to the early postpartum or were long-lasting, what we did was to scan a subsample of these mothers 2 years after childbirth. And what we found is that most of these changes are still present 2 years after parturition. Okay, so we found that pregnancy entails long-lasting gray matter volume reductions in regions of the default mode network. But what does that mean? What is the significance of these gray matter volume reductions? Are they related to cognitive performance, to what is commonly known as mommy brain or mommynesia? So in this study, we really wanted to see if there were differences in cognitive metrics. So we obtained cognitive metrics before the mothers got pregnant and then again after parturition. And we did not find any differences in cognitive measures, nor did we find associations between gray matter volume changes and cognitive measures. What we found were indicators that gray matter volume changes were related to maternal behavior and maternal attachment. And let me explain that a little bit better. So on the post sessions, once they have the baby, we ask participants to respond to a questionnaire that measured maternal attachment. And we found that the more the brain changed, the higher the scores on maternal attachment scales. Also, on the post-session, mothers perform an fMRI, that is a technique that measures how the brain is active while the person is doing certain tasks inside the scanner. In this case, the task was that mothers have to view pictures of their baby. And we found that the regions that were active while mothers view pictures of their baby partially overlap with those that present gray matter volume reductions. And this suggests that the changes are directed toward facilitating maternal behavior. Okay, so therefore, on the question of whether pregnancy changes the human brain, the answer is yes. Pregnancy produces consistent gray matter volume reductions in the mother's brain that are associated with maternal attachment that persist at least for 3 years. But the results of the study also raise a number of questions that we have been trying to answer during the last 5 years. And in the course of the rest of the talk, I will briefly walk you through all of them. Let's start with the first one, which is, are the changes in the brain similar to those in adolescents? Why are we asking that question? So the thing is that when the results reached the press, some journalists made statements such as, woman's brain atrophy with pregnancy. And when they interview us and we have the chance to respond to that, what we did was to explain that a reduction in gray matter volume does not necessarily imply a neurodegeneration, and that in biology, as you know, sometimes less is better. And to explain this, what we did was to use as an example what happens during adolescence, where these hormonal changes are associated with a reduction in gray matter volume, but also with a maturation in cognitive function. So we thought that the changes we observed during pregnancy were similar to those that happened during adolescence, but the truth is that by the time the first study was published, we hadn't empirically tested that. We did so later, years later. So specifically in 2019, we conducted a study that compared the brain of girls before and after puberty with the brain of mothers of women before and after their first pregnancy. And as in the previous case, we also include a control group of nulliparous women. For this study, we quantified a very detailed number of morphometric features such as cortical volume, but also cortical thickness, surface area, whole area, gyrification, and so on and so forth. All the ones that you can see here in this image, which give us a very detailed description of how the cortex is. Here, I show you the main results of the study. So in this figure, what you have is the percentage of change in the y-axis, and each of the brain matrix, each of the brain measures that I told you about on the x-axis. And the horizontal line just displays the zero, that is the no change. In different colors, you have in blue the mothers, adolescents in red, and female controls or nulliparous women in green. And as you can see, the profile of neuroanatomical adaptations in mothers was identical to that observed in the group of female adolescents. And what these results reveal is that both pregnancy and adolescence entail a comparable flattening of the cortex. Okay, another question we had was, do human pregnancy also modify subcortical structures? Why? Well, so far we have been mainly focused on the neocortex, which is the newest part of the brain phylogenetically speaking. So we wonder whether older or more evolutionarily conserved structures that control maternal behavior in rodents, for instance, the nucleus accumbens, do also change in humans. So we were specifically interested in this region, the nucleus accumbens, as it is a key area to signal the paps as rewarding for the mother. And we want to know if this signaling of rewarding, this motivational circuit also changes in human pregnancy. What we did was to manually segment this brain area in the group of mothers and nulliparous women that I have told you about. And we found significant volume reductions in the right accumbens in the mother's group. And interestingly, the more the volume is reduced in this region, the greater this region activates when the mother saw pictures of her baby. Sorry. Next question we had was, do pregnancy-induced brain changes reverse? So from the previous publication, we found kind of this trajectory with decreases during pregnancy that are still detectable at 2 years postpartum. But on the other hand, there are researchers performing big data cross-sectional studies that compare the brain of thousands of mothers to that of thousands of non-mothers at midlife. And these studies found younger estimated brain age in mothers versus non-mothers. And what this suggests is that pregnancy has a lifelong impact on the brain. However, this is only indirect evidence from cross-sectional data, and we need longitudinal evidence to really test whether the changes induced by pregnancy persist beyond the 2 years postpartum, which is the period of maximal maternal investment comparable to that to weanings in rodents. And therefore, to fill this gap of knowledge, we scan those mothers from the initial study that had not been pregnant again at 6 years postpartum. And what happened here? So the normal thing is that some of the mothers already had a second child, so we discard them, and some of the controls got pregnant, so we discard them. And what happens is that we end up with a very, very small sample size. But I want to highlight that despite its very small sample size, so far it represents the only available longitudinal data to examine the long-term effects of pregnancy on the human brain. Okay, so this figure displays the gray matter volume changes at the whole brain level. Here I paste the gray matter volume reductions reported in our previous publication just as a reference. So this is gray matter changes from preconception to 2 to 3 months postpartum. And this one here is gray matter changes from preconception to 6 years postpartum. And as you can see, key areas of the deformal network, such as the medial prefrontal cortex and the precuneus, remain reduced at 6 years postpartum. And when checking for each of these regions separately, we observe the same pattern. That is, here you can see mothers in black and nulliparous women in gray. And what you see is that at 6 years postpartum, gray matter volume reductions in these regions remain reduced in the group of mothers. Well, next question. This is one of the questions that everybody asks me when I go to conferences. What happens in the father's brain? I'm sorry, but I'm not going to go into the detail or I'm not going to expand very much on this point, as I know that other researchers are going to address this topic, and it's not the core of my talk. But I will, as I told you, I will just dedicate this slide to talk about them. Well, as you might remember, when I showed you the results of the first study, what I told you is that we did not find any significant change in the father's group. And I want to highlight the significance, because there in that study, we use a very restricted threshold to determine what was significant and what was not. But when we plotted the raw data, like the raw change, what we found is this type of distributions. Here you have the distribution of gray matter volume, thickness, and area. In pink, you have the distribution of mothers. In blue, you have the distribution of controls, which is centered on the 0 of no change. And in orange, you have the distribution in fathers. And as you can see, the distribution of changes in the fathers group lays in between the other two. So in short, father's brain changes, but the changes are less pronounced than those observed in mothers and seem to be more variable. And as in the case of the mothers, brain changes in fathers seem to be associated with different aspects of paternal behavior, such as father-infant bonding, and also with the father's response to baby stimuli. So here you have the title of the articles in case you want to take a look at them later. Next question, which is the temporal course of the brain changes? So far, I've told you about the longitudinal perspective studies that follow the brain of women from before conception to after birth. And these studies report consistently gray matter volume and decreases during pregnancy that are still detectable at 2 or 6 years postpartum. But there are another set of studies that follow the brain of women longitudinally during the postpartum. And when analyzing exclusively the postpartum period, these studies report either no changes or increases in gray matter volume. So how do we fit all these findings together? Well, by pulling together all this data, alternative trajectories emerge, including, for instance, this one over here, in which it is possible that the decreases we have reported in our previous publication specifically occur at the time of birth and then recover a little bit during postpartum. But that's not the only trajectory. I mean, we can think about as many trajectories as potential combinations you can imagine by connecting these different points that it is actually where we have measures. Well, so to better delineate this trajectory, what we did was to perform a study in which we zoom into the unexplored period of the peripartum. And in the paper I'm going to tell you about, which is currently under review, I'm not going to go into the details, but I want to walk you through it very briefly. So what we did was to scan 110 primigravid women at late pregnancy, and then again during the first month postpartum. And as in previous cases, we included a control group of nulliparous women scanned at similar time intervals. Okay, for simplicity, I'm just going to focus on cortical thickness, but I can already tell you that the results were very similar also for volume and surface area. What did we find? So when we compared these two groups at this time point, pregnant and control women cross-sectionally, we found that the pregnant women show marked cortical thickness reductions even before birth. And some of these reductions were still detectable when we compare cross-sectionally these postnatal sessions. What this suggests is that reductions are present before birth and are still detectable during the first month postpartum. Then we also performed longitudinal comparisons between these two time points and also taking into account the control group. And we observed that there were increases in the group of mothers as compared to the group of nulliparous women. Furthermore, these increases were positively associated with postpartum duration, suggesting that birth is the inflection point in these gray matter trajectories. Then we also have another question that I really find very interesting and a field to explore in more depth is regardless the birth type. So as you know, many women have a vaginal delivery, but there are another percentage of women that had an emergency C-section or an scheduled C-section. And we want to know if there were differences in the percentage of change between women that have a vaginal delivery, emergency C-section, or a scheduled C-section. So here I show you the percentage of change for the three metrics, that is volume, thickness, and surface area, and the four groups, which are controls, vaginal delivery, emergency C-section, and scheduled C-section. As you can see, we did not find differences as a function of delivery mode, that is, differences between vaginal delivery or C-section delivery. However, we found differences between those mothers who experience the first stage of labor, that is, experience the intense uterine contractions and cervical dilatation, and those that did not. In other words, we found difference between mothers who gave birth by scheduled C-section and those who experienced the first stage of labor, that is, vaginal C-section or emergency C-section after labor. In particular, what we found was that the percentage of cortical increases between these two sessions is more pronounced in women who had scheduled C-section than in women who initiated labor, regardless if labor ended up with an emergency C-section or a vaginal delivery. And after some extra analysis to discard other potential confounding factors, what this data suggested is that parturition itself, the process of labor specifically, also entails neuroplastic mechanisms in the brain that translate into further cortical decreases. Okay, so far, to sum up, what this data points is to a trajectory similar to the one I'm drawing here with gray matter decreases during pregnancy that not only valley at parturition, but accentuate during labor and later revert as postpartum progresses, but do not reach pre-pregnancy levels, at least in certain areas, such as the deformal network. Well, just to sum up what have we said during the talk, we wanted to answer several questions. The first one was, does pregnancy change the human brain? The answer is yes. Pregnancy leads to long-lasting gray matter volume reductions in regions of the deformal network that are associated with different aspects of maternal care. Then we also wanted to know if the brain changes we have observed are similar to those taking place during adolescence. And we found that yes, both pregnancy and adolescence are associated with a comparable flattening of the neocortex. Another question was what was happening at the subcortical level, at these core regions of the non-human animal maternal circuit. And we found that these subcortical areas are also reduced in women, especially the nucleus accumbens, which is a key node on reward processing. And the greater the volume reduction in this region, the greater the functional activation of this same area when mother saw a picture of her baby. We want to know if the brain changes reverse or are they lasting. And the truth is that we don't know yet, but it seems that at least for certain parts of the brain, especially the deformal network regions, the changes seem to persist for at least 6 years postpartum. The question about the fathers, do father's brain also change? Yes, father's brain changed, but the changes are less pronounced than those observed in mothers. We also want to know about the time course of the findings, and I show you about this publication we have now on the review that shows a trajectory compatible with gray matter decreases during pregnancy that accentuate during labor and slightly recover as postpartum progresses, especially during the early postpartum, but do not reach pre-pregnancy levels, at least in the deformal network. And that's all for my part. I want to thank, well, as you know, all these studies have been the effort of many, many people, and I want to thank all of them. But I especially want to thank the work of my past lab mates, especially Elseline Huxema, Erika Barba, and Oscar Villarroya, as well as the work of my current lab members, specifically Magdalena Martinez, Maria Paternina, and Daniel Martin de Blas. I would also like to take this opportunity to tell you that if you're interested in keeping up with our publications, you can visit our website, Neuromaternal, as well as follow us on social media. Well, thanks a lot for your time. And if you have any questions, I'll be happy to answer.